I'm thrilled to invite Jennifer on stage. Jennifer is our keynote today, and she's a professor in the Department of Electrical Engineering and Computer Science at the Center for Computational Biology and the Bioengineering Program at the University of California, Berkeley. I think we all know a lot about Jennifer already, so I'll just give her the stage, and please. Thank you.
Oh, I feel privileged. I don't have to knock on the door. This is great. So it's a pleasure to be here. And I think I only have a small part of my talk that will piss Pat off. So we'll see. Where is Pat? Very good, very good. It's a little like NeurIPS-y indulgence. It's not in NeurIPS, but okay, so I'm gonna just kind of have 4 parts, sort of a general overview of what I think people are thinking about and doing in the space of machine learning, or now AI, and protein engineering. Then I'm gonna give an overview where I'm just gonna tell you a flavor of some of the things we think about and the challenges. And then the third one is the one that will piss Pat off, which is just a little bit about a particular nugget of a problem in diffusing on discrete spaces that I'll explain. And then I'll go more detail into one on re-engineering AAV for therapeutic use. And I'll probably at the end skip a few slides because I've injected some new stuff, but it'll all work out. Is this? Oh yeah, there we go. Okay, so I don't think I need to argue to this crowd why it's important to be able to make further advances in protein engineering. It touches not only on drug therapeutics, but also on agriculture and environmental kinds of problems. And so if you just put on your kind of abstract computational thinking hat as a computer scientist, you realize there's just a huge combinatorial space. So even if I have a protein that's of length only around 50 amino acids long, the number of combinations is already getting near the number of atoms in the universe. And so forget the fact that I don't even know how to measure things that I like at a high scale and so forth. Just like at some very fundamental level, this is a huge combinatorial space that we need to search through and find what we want. And so, of course, there's a precedent for doing this. It's called natural evolution. Has given us, it's why we're all sitting here with all these lovely proteins in our bodies that let me give this talk and breathe the air. But it takes so long that the purpose of protein engineering is, of course, to accelerate that into something finite in our lives. And so there have been these 2 main ways that people are doing protein engineering, and only until recently one of them had a Nobel Prize. Now both sections get a Nobel Prize. And so this sort of data-free approach is this sort of Rosetta biophysics-based approach that Baker drove forward for many years, and it says almost rest in peace. So there are a few corners where people claim that this is still quite useful above and beyond the machine learning approaches that have taken off. And then there's, of course, the wet lab of directed evolution. And the reason we're all here together and I'm talking to you is because machine learning is interleaving with all of this. Sometimes people pretend it's taking it over, but of course, we still actually rely on combining many ideas from biophysics, from data from the wet lab, and machine learning in order to have some successes. So when I tell people that I work on protein engineering, which I started to do about 7 years ago, people say, oh, didn't AlphaFold 2 solve your problem? And so I just want to emphasize that AlphaFold 2 was a tremendous success. It's an incredibly powerful tool, but it goes from sequence to structure. And if I were to want to use it for protein design, if anything, I would need it to go from structure to sequence, because protein design is about, I tell you what sequence to make in the laboratory, right? Nothing else works. I can't tell you the structure, can't tell you the function. I need fundamentally the sequence. And so there are these inverse folding models. So my student Chloe Su, actually at Facebook, basically trained AlphaFold in reverse at scale to get one of these inverse folding models. And those are a useful component of machine learning-based protein engineering, but they don't solve the problem. And something that's overlooked in a lot of the fancy machinery of generative models and so forth is that we don't actually know the link typically between the structure or the sequence and the function unless it is structurally mediated. It's like a lot of binding maybe, but for example, catalysis, we don't know how to change like an active site to get an enzyme to do what we want. And so when I first started working in this area, I was very confused. There would be a new paper out every week claiming to have solved protein engineering, and it was all sliced and diced in strange ways. And so one of my students kind of put this idea together. You could divide the AI task into some prediction tasks. And this is a little arbitrary, but I think it's helpful to think of this way. So machine learning prediction tasks, and then the next slide will have what I call design tasks. So the prediction tasks would be, I give you the sequence, you predict the structure. That would be AlphaFold. Or I give you the sequence, you predict the function. or I give you the structure and you predict the function. And so people are working on all three of those problems. And then there's going to be three more on the next slide. And altogether, these are the kind of little chunks of the overall problem of protein engineering that many people work on. And then we sometimes assemble them together into some pipeline. So in terms of design tasks, fundamentally, we want to design the sequence. And it'd be great if we could just specify the function and design a sequence and be done. We can't generally do that very well. Another task we might set ourselves is to say, I condition on a function, and you tell me the backbone structure, and then given the backbone structure, then you generate the sequence. And then I'm piping together two different models. And this is actually one of the ways that people do things right now. But they can kind of, as I said, only do structure-based function. They can't say, catalyze that reaction, go. This is just not possible at the moment. So that's like an overview of some of the types of problems, both prediction and design, that all get used in a basket together to try to do the overall. And of course, protein engineering means very different things depending where you're starting from, what throughput of data you can measure, what's already known about the protein, and so forth. But generally speaking, there's a few trends in how people use machine learning to advance the field of protein engineering. And a really big one, for example, Facebook and now the sort of spin-off company that Alex Rivas has been heading, are doing a lot of representation learning. And so for those of, and that's a sort of buzzword, but a real world in machine learning in all kinds of areas, not only in the sciences. And really what it means is that to give data to a computer, it has to be in numeric form. And if it's not in numeric form, if it's in a graph form or it's in a sequence, I have to get it into bits. And you can do that with sequences of amino acids very trivially through some just deterministic encoding. But these encodings are not sort of the most useful. And the idea of representation learning is to take a huge set of, say, protein sequences and learn a numeric encoding that understands something about sequences such that I can use that encoding for other problems that have less data and therefore get better statistical efficiency is the hope. Okay, so that's representation learning. And you may do that, and then a second, really, this is like sort of a hot topic, is conditional or generative models and conditional generative models. We all know that ChatGPT is a generative model that does next token prediction. Similarly, you can have a generative model over protein sequences, or and you may condition on the structure. So you may say, give me, this is the backbone structure I desire. Please tell me the amino acids I should use or a distribution over them. Or sometimes you may condition on the family. In a perfect world, you could just condition on the function. But again, to do that, it's not sufficient to have fancy machine learning machinery. You need to have the information that actually links these somewhere in there. And then another thing that people have spent a lot of time are these generative models for the backbone structure. And there's one from David Baker's group, and then there's John Ingraham et al. That sort of did it at the same time. And these are using these sort of well-known diffusion models that Pat alluded to. And again, these are like really elegant and they can be very powerful, but you have to fundamentally know something about the function in order to use them properly. And again, if you don't have that information that links the structure to the desired function, then all that fancy machinery is not going to buy you anything. I'm not a downer on this. I just think this is overlooked. I think these are powerful, interesting models that are already probably maybe useful. And if you want an overview, sort of primer of some of these ideas of generative models, you can check out a recent primer that my students and I wrote, posted here. And then maybe this one is also, this is one of the least sexy problems, but very important problems, is how do I estimate some link between the sequence and the function so that I can know about that how to engineer for the function I desire when it's not just structurally mediated in particular. And for many problems, when we start an engineering campaign, we have few or no labeled data. And so we rely often on the kind of information that AlphaFold also relies on to do structure prediction, which is evolutionary data. So just as AlphaFold doesn't work well if you don't have a set of homologous proteins to help inform the structure prediction, similarly, if you don't have homologs to start off an engineering campaign, then you're starting off kind of with zero data. And so we really do leverage this evolutionary data to help us start a campaign that will be iterative and we gather more information, but it helps direct us to start the first round of experiments. And then, of course, like AlphaFold 2 was amazing, and AlphaFold 3 might be, but as Pat said, we don't completely know because we can't access it. But I think some of the really important problems in any case, whether we had access fully to AlphaFold 3 or not, are this idea that actually we really kind of want to know the main conformational states that a protein takes on. Maybe we sometimes want to know the dynamics as well. And fundamentally, we just can't do that right now. Like the closest way to do that is basically molecular dynamics, and that doesn't work particularly well and is very expensive. And so this is one of the wide open things that people can't really tackle that is felt would help improve the successes in this general area. And so sometimes I've gotten into a surprising number of arguments with people that now that we have generative models, we can just generate more data so that we can get the models that we want. Because one of the problems here is we just don't have the data to get the dynamics. And so I woke up one Sunday a little annoyed with telling people over and over we can't just fill the data gap by using generative models, which is to some people strikingly obvious and to others strikingly not obvious. Anyhow, you can check this out if you want to hear some of my thoughts about the hype in AI and science and why structure prediction in particular was a task that could be solved well, static structure prediction. Okay, so that's sort of loading you up on what's happening in this field in general. And so now let me give you an overview. I try to find the areas that aren't too crowded that I think are impactful. And so on the one hand, we do some methodological development just on our own. I don't run a wet lab, and then we also collaborate with wet lab investigators to keep ourselves kind of honest and working on real things. But what I'm going to tell you now is like a little vignette about my entryway point to the field of protein engineering, which I got into by accident. By thinking about a certain kind of problem that ended up looking a lot like this. So in standard supervised machine learning, you take a set of X's, like a protein sequence that is paired with a known label. This is multiple labels. You can just think of one label, like this is sequences, maybe this is GFP fluorescence, something like that. And then because they're paired, your goal as a machine learning person is to set the knobs of your predictive model, here a neural network, such that on the training data, you map the X's to the Y correctly, and you have to be a bit careful so that it works on unseen data and doesn't just memorize your data. And that's basically the game of supervised prediction. But when we do design, what we're fundamentally doing is saying, if we do machine learning-based design, then we assume we've sort of encoded some knowledge in a network that maps something about the sequence to the function, and then we want to invert the model. So we don't want to adjust the parameters anymore. We assume we've done the best we can at learning this functional mapping. What we want to do now is specify a property like higher fluorescence than I ever saw in the data set, or better catalytic efficiency than I saw in the training data set, leave these knobs fixed, and actually optimize for the sequence that will give me the most highest value according to this neural network, because that's kind of the best I can do if this is the approach I'm going to take. And so this is actually much, much, much harder than supervised learning for several reasons. And so let me just give you one sort of, it's my banana analogy. So it turns out in computer vision, people have something that smells a lot like machine learning-based design in the way I just presented it. And I should say, you may think, oh, well, generative models don't behave like this. But under the hood, everything has this problem that I'm about to show you, whether it looks like this or it's an actual generative model. But you specify, so the machine learning people take this neural network, they train it on many, many different classes of objects, banana, kiwi, car, whatever, and then it's a state-of-the-art classifier, and they say, I want to understand what my neural network thinks a quintessential banana is. So similarly to how it might say, give me the highest catalytic efficiency, they say, turn the banana node to 1 and turn the other class nodes to 0, leave the parameters fixed, start with a random image, and iteratively optimize it till I have the most banana-like image. And so when you do that, you get abstract art. And so, if you were to try to go print this and put it on the table, you couldn't, right? It could just be a 2D thing. You couldn't make a 3D object of this, even though it's plausible, it has the right curvature, the right colors. And so by analogy, this is actually what happens if you try to do machine learning-based design and you try to squeeze the network for all it's worth, and you're squeezing it outside of its comfort zone is really what's happening. Is it only understands things near the training data, and when you try to do what I call extrapolative design, it's almost certainly going to fail and just give you nonsense. And so this is the sort of the initial, this set the stage for the initial set of work that my group did when I started to work in this area, which is fundamentally navigating this tension between how can I extrapolate from a learned function that tells me about the property I care about, but have to understand that that model I can't trust it everywhere. And there's fundamentally always going to be such a tension if you're trying to do this sort of extrapolative design. If you're trying to design things within the scope of properties you've already seen, then I think you have a much better chance, but often that's not what we're doing. So there's, I'm not going to go into details, but some of our earliest papers are trying to basically get at this either by by just pointing out the problem, because people were just doing optimization, saying they were doing protein engineering, or trying to really get at technical ways to help understand where can I trust the model and how far can I go before I should collect a new round of data, which is a bit of a risk assessment, and you may have different risk for different problems. And maybe I'll just highlight this one paper that Hunter Nisanoff led and came up with the idea for, which is to say, we know that machine learning models typically outperform biophysics based models that may exist for the same problem near the training data. But as I go further from the training data, like all bets are off for the machine learning model, whereas since biophysics-based models typically only at most tune maybe a couple of scalar parameters on real data, they should be kind of equally good across the whole space, but worse than the machine learning models near the training data, because machine learning is powerful if you use it properly. And so in this paper, he says, let's get the best of both worlds, where we understand as the machine learning model kind of degrades and becomes more and more uncertain, let me seamlessly blend the predictions from the biophysics model sort of gracefully as I go further away from the data. I'm not going to say much about the other ones in the interest of time. But if you're interested in this sort of whole space, also Clara and I wrote a sort of perspective / tutorial weird piece that is basically talking about all of these problems of how do I do extrapolative design? Can I actually design for novel properties using machine learning? Like, how do I think about that? And it's a general framework of a discussion, but it's anchored in protein engineering. Okay, and then so some of the other topics that we've touched on include the fact that often in the sciences, I mean, it's less so now, but people will just take existing neural network architectures like convolutional neural networks, transformers that were, in particular, let's say convolution was designed for images, like there's something about spatial smoothness and so forth. And so this is what we call an inductive bias in machine learning. It's putting in some of your own knowledge into how you design the architecture of the neural network. And it's not so much the case now. People are thinking about graphs and other things, but often we just grab whatever worked in computer vision or natural language processing and directly apply it to our scientific problem without thinking about what those assumptions were and if they make sense. And so we have one is a collaborative paper with Amirali and Information Theory Group, and we have a follow-up that's not yet out. But the idea here is to say, if we're making functions, trying to estimate functions that go from sequence space to a property of interest, then I should think about inductive biases in the epistatic landscape, which is sort of the complexity of the interactions between the sequence positions. And we know that most biology is very kind of low complexity on that scale. It's often linear, additive in positions. Sometimes there's pairwise. And the higher I go in the order of the epistasis, probably the less likely it is. And so the idea is, can we encode that into our neural networks? And so now, and then this is also an accidental paper. So what we set out to do here is some of the papers that were coming out in the early days of saying, how can I do low-end protein engineering? I have just a few supervised examples. Can I design for a property when I have only a few supervised examples to train my model? And can I use large unsupervised data sets to combine with them and do better? And these were creative, interesting papers, but we couldn't tell if they were just interesting stories or they were real results. And so we started to compare. And this is a little bit outdated now, but so we ended up with this big comparison And when we published this, they wanted to highlight our new method, but the point of our new method was that it was really dumb and simple and beating the really fancy big machine learning models. And so I think that was a lesson that is often hidden in a lot of these papers, that simple things without these big models actually work really well. And also, in fact, there's something fundamentally wrong with this paper, and many of these papers, is they all work on a bunch of data sets that are looking only at 1 and 2 mutations away from a wild type. But the whole point of machine learning is to do more than that. And then we sometimes benchmark on these data sets, and we did because that's all we could do at the time. It's becoming a little bit better now. And these two I'm going to tell you about in more detail, so I'm going to write in a moment. But right, so sometimes we have these kind of, you can see maybe pie in the sky, maybe a bit more theoretically oriented projects that we think are useful to help clean up some of the thinking and that will lead to hopefully practically useful algorithms and models as well. But we like to work very closely with people who have a wet lab to make sure we're working on real problems and maybe pivoting or tweaking where we were in our computational lone heads. And so there's a number of them, and the one I'll talk about in a second is the first one with David Schaefer, which is the most mature of these and is already published. But I like to put my acknowledgements to my group in the middle so I don't get stuck without them in the end. So I'll just say these are the people of the work I'm talking about today that made it happen or are making it happen. So in the next 2 projects, I'm going to talk about David and Akosua, who are now at Dino, which is here in Boston. I think neither is in Boston. And they led this AAV project. And then Stefan, Hunter, and Bear led I'm going to tell you a little nugget of a project that's a bit about some diffusion models. And then Chloe is the one that did that kind of big comparison and showed that a simple model worked. I think those are the main acknowledgements for this talk. Okay, so this is, I just sort of inserted this in today. Pat, you can close your ears now. So because this is going to end up talking a little bit about diffusion, but I'd like to set the stage here, which is, here's something that happens a lot in machine learning-based design, is I have a generative model for something just like sequences, all sequences, and I can build such a model kind of fairly easily, because if all I want to do is generate a plausible protein sequence that's likely to fold and be stable, then I have tons and tons of proteins in UniRef, and I can train a big unsupervised model on these, and that's pretty easy. Of course, no one cares about just randomly generating a protein sequence, right? What I want to generate a protein sequence X conditioned on some property that I care about. So I want to do conditional design. And so what we usually do, not only in the sciences, but this is even what ChatGPT does, they take a big model and then they kind of refine it in some way to get it to do what they want. And this can be, people use different words. Technically, what you're really trying to do is take an unconditional model and make it into a conditional model for some property you care about. And so the question is, like, how can I do that? If I have this P of X, I've trained it, and I have either a predictive model for the property I care about or a set of data, how can I adjust, like, given that I put all this work into P of X with a ton of data, how can I kind of squish P of X around to get the correct conditional distribution? And this is, you can think of this both for proteins, there's sort of an analog in the small molecule space. This is a problem that kind of appears everywhere. And so there's, and also people like don't really, I don't know why, they don't talk about this. They just present one of these as though this is obviously the right way to do it. And I think it's interesting to ask which way provides, what are the pros and cons of these different methods? And I'll talk a little bit about that. But the first thing you can do is just kind of actually bake it in, which is to say you don't start with an unconditional model. You just start with all the X's and Y's you have access to, and you just directly build this conditional model, and then you sample X conditioned on Y. This is not very commonly done. Another thing you do is you start with that P of X, this unconditional model, and then you use the predictive model or the data and you update it. And you have to retrain the model. It has a new set of parameters, and now you have this one static model that you can sample from for the condition you care about. And the third one, and I feel like John Ingraham in his Chroma paper highlighted this particularly nicely, which is that you can also take this P of X model and not change it at all, but play like Lego blocks with your like a classifier over the property you care about. And in the parlance of machine learning and these diffusion flow models, we talk about guiding the model. And so what's nice about this, it is very plug and play. You can put all this energy and all this data into an unconditional model, get it to be as good as possible, and instead of having to like retrain it for each new condition, you can take your classifier, and as you're generating a new sample, they kind of play together to generate the right kind of sample. It's a very technical area, and I'm just trying to give you a flavor here of how it works. And so this is, I would say, one of the most popular ways, in particular because in the sciences, we do often have a lot of unsupervised data, and we want to put a lot of these domain-specific inductive biases into a giant P of X model, and then we want to be able to reuse it to do different things for different conditioning exercises. And so who knows Bayes' rule? And I get a read on the room. Whoa, all right. A lot of people know Bayes' rule. If you don't worry. All I want to make a point here is that if you have an unconditional model and you have a classifier for the property you care about, then in order to invert the order, what you really want is you want to generate sequences based conditioned on a property. You have to use this Bayes' rule, which is just this very simple but very powerful, widely used equation. And so I actually don't understand this reference. My student did it. Does somebody get it? It's some popular culture that they always have these jokes. I don't know what they mean, but people laugh, so I left it. But the problem is, like, so I understand it's bad. This is bad, and that says down bad, so I'm happy to keep it on the slide. So this is basically generally intractable. Like, we cannot deal with this, and if you don't know why, don't worry, I'll just tell you. And so what that means is if I have a generative model that's unconditional, and when I combine it with a classifier, I have this huge problem. And so diffusion models are very nice. Again, don't worry about all the math here. I'm just going to tell the story very simply. But what happens in diffusion models is you do this thing where you actually use the gradient of the density that you care about. Instead of getting just this thing, you want to take the gradient of it. And once you have that in your hands, you can generate samples by following a procedure. And what's nice is when you take the gradient of this whole thing, don't worry, what happens is that term that we don't know how to deal with, it just falls out. And this is one of the powers of using guidance in diffusion models is we can just get rid of that nasty term. So, however, all the work in diffusion and flow models for a long time was just in real valued spaces. So on like things like images, which are actually discrete, but we think of and treat as continuous. We care a lot about designing sequences, or you may care about designing small molecules, and these are fundamentally discrete objects, right? Like, it's very different to generate a real valued, say, 3D structure or image than it is to, say, generate a sequence of things. And we know, for example, ChatGPT generates a sequence of things, but it doesn't use these very powerful models at the moment, these diffusion and flow models, which have really shown to be incredibly useful especially in computer vision. And now, I was actually quite skeptical that they were useful on discrete state spaces, and I kept telling my students, so it's totally different, why do you want to use these models here? But they eventually convinced me, and that's another topic of discussion, is like, why are these useful? Because it's very different. But what happens is it's so different, in fact, that you literally can't even use the same underlying algorithms, models, probability, anything. Everything falls apart. And the reason it falls apart is when you go to a discrete space, all these gradients don't exist, basically. Like, you can't compute them, or you can't get information from these gradients. And so all the underlying machinery is broken. And so there's a fantastic set of work by Campbell et al. In particular that gets around this by using a very technical machinery that I'm not going to go into called continuous time Markov processes. And I think that's the most elegant solution to do diffusion over discrete state spaces. And so what they didn't do, what was missing, was how can I generate in a conditional manner? How can I condition on the property I care about? And there's no point having a fantastic generative model if you can't condition and on the property you care about. And so basically my students have filled in this gap. So Hunter, Bear, and Stefan led this project. And again, it's quite technical, so I'm just trying to give you an overview of the main idea here. But fundamentally, you replace these gradients with something called a rate matrix here. You don't really need to worry about the math. And just simply by going into this continuous time, what happens is I know you're not going to get it here, but you could look in the paper if you want. But you get around the problem of the normalizing constant at the same time as being able to kind of correctly do diffusion in these spaces. And so this was something people actually have been trying to do, saying, I don't know how to do guidance on these kinds of models. And so now you can do it in a there's a lot of derivations here, so it's a very clean system that they've derived where you really can do this with the correct formalism now. And I mean, I don't know who. Let me just take a poll. Who in the audience does this line mean something to? And if it's too few, I'm not even going to talk about this slide. Okay, do a few, just like a few, I'll very briefly. Okay, and the rest of you just tune out for 10 seconds. So these are with in regular continuous space, you get the conditioning distribution gradient that you want by taking the unconditional one and adding the gradient of the classifier. This is called classifier guided generation. And so basically what my students show, it looks very simple here, but again, it took a lot of cleverness to actually figure this out, is you get a very analogous equation. And basically the core entity in these models on discrete spaces becomes this rate matrix instead of the gradient. And they show that you get a very analogous update. And so you get a very similar ability to plug and play very nicely. And I'll leave it at that. And this was one of these sort of toy NeurIPS papers just to bring an idea into the world. So I actually think, I love NeurIPS in the sense that I take NeurIPS as a publish creative, interesting ideas and don't believe the results venue. But we have to put the results in there so and I think it's not a bad idea, you just shouldn't take them too literally, right? So I'm not claiming we've solved some new problem here, although I think the third one actually is quite interesting, and if we pushed it further, it would be potentially solving a real problem. But we basically applied this diffusion over discrete state spaces for small molecules on a particular toy problem we don't really believe in, but just demonstrates the machinery works, one for some enhancers in DNA sequence. And in this case, Hunter actually trained his own inverse folding model and showed how to use this machinery to generate things that would be sequences that fold just as well as a regular inverse folding model, but be more likely to additionally be predicted to be stable by guiding it to be more stable. Okay. So now I'm going to go into for those of you who found that too technical and weird, hopefully you can enjoy this part a little bit more. This is this project took many, many years. It finally came out earlier this year, which blows my mind. It feels like it should have come out a long time ago. But so David Baker is a colleague of mine who's a world expert in directed evolution. And in particular, one of the things he's focused on for a long time is using this adeno-associated virus as a delivery mechanism for therapeutics. And this is the team of folks that have worked on it. And so this actually has been very promising. There are clinical trials that have shown this to be useful, and you probably have read about it in the news now. There's actually, this is really happening where people are getting a real win from delivering something within AAV. But the problem is that all of them basically have to be behind the blood-brain barrier, so the antibodies don't shut it down. There's a bunch of cells we don't know how to target. We don't know how to change the AAV virus to target the cells we want and not to target the cells we don't want. So there's although it is still it has had some good successes, there's a long way to go if we want to use it more generally. And so what this project was about was what I call, from my perspective, I think an understudied and really lovely problem for machine learning people to think about is library design. When I'm doing either just from the get-go, what's the first library of proteins I should measure in the lab, and then iteratively, how should I go about doing that? And you could think of that as classical design of experiments, but I think in the era of machine learning, there's a lot of kind of interesting problems that get folded into there. So the goal was to say that Dave does all these different campaigns to try to change AAV like this or like this or like this to achieve different things. And every time he does it, he has to kind of start from scratch with what's essentially a uniform library of proteins. And so let me clarify what I mean by uniform library of proteins. So in this case, one of the things that's got him a lot of wins is in his directed evolution, you need a form of, you need some mechanism to induce variety to do the evolution. And one of the ones that's very powerful in AAV is to insert a 21 nucleotide insertion into a very specific part of the capsid. And this produces a tremendous amount of variety in many phenotypes. And so this is something that they often start a campaign with, is they just say, you know what, for this 21 mer nucleotide, let's just make random 21 mers uniformly and insert them at scale, something like 10 to the 6, and then do directed evolution. But when you do that, although this is a powerful mechanism, what happens with those libraries that start sort of at uniform, it's not quite uniform. It's technically called NNK to reduce stop codons. But if you don't know what that means, you can just think of it as uniform over the space of nucleotides with a small tweak. And when you do that, you waste half the library from the get-go. And the reason you waste half of it is half those viruses just don't package. Like, they're basically not functional. And the game of directed evolution is a game of getting yourself the best lottery tickets possible. And so here we know that half our lottery tickets basically are failing from the get-go, and they have no hope of helping us in our directed evolution experiment. And you can filter them out, but you're losing those tickets, so you're losing like your shots on goal. And so the idea was, can we create a better starting library for any downstream task that Dave may have in his lab, any engineering goal for AAV? And so what does it mean that it's better? It means we have just as much, ideally just as much diversity as uniform, or close to that, but have better packaging efficiency on the whole for the whole library so that we get to keep all our lottery tickets. And so this is what we set out to do, and this is what we have done, and this is now one of the libraries that he uses. So the first thing we did is we took his NNK library, which came with sequences and some log enrichment. I could tell you, there's a whole paper about, not how we did it in this paper, but I'll give you a teaser if there's time. But we built a supervised model from sequence space of 21 nucleotides. Actually, I think we did it at the amino acid level, to the packaging efficiency for the library he was already using, this uniform or NNK library. Don't worry about the details here, but basically we pick out a neural network architecture, very simple one, quite small, that just does best at predicting packaging efficiency from sequence on the NNK library. And then we wanted to check how well this model worked, so we picked 4 variants, 5 that we had not seen in the training data, including the global predicted maximum, and then sort of like a dilution series, and show that this model actually works really well across the whole range. So this model actually understands the whole scope of this problem. We don't have a lot of extrapolation issues here. We just want good packaging, and we have seen some good packaging, and the model works in the whole range. And so now this is sort of a simple idea here, but one that's missing from a lot of machine learning library design. So what you almost always have some sort of trade-off when you're designing. And what the trade-off we have here is, each point here is a library. This is a machine learning library design D1, machine learning library design D2. This X here is the NNK library. So every point is a library, and each library specifies, I think I forgot to mention this, it specifies the probability of ACTG at each of the 21 positions. So instead of uniform, I can make it non-uniform, but I can only control it at each position separately. But that's still quite a bit of control. And all the machinery I'm telling you about can be extended when you have more control. If you could do full synthesis, you can still do what I'm going to tell you about here. But so in our case, this NNK library, it has the most, of all the libraries we computed, it has the most pairwise diversity. And that's because it's very close to uniform. And on this side over here, if you take this library, don't worry about the spread. This should just be a clean line. It's an optimization issue. But if you take let's just say this library here, this library has really high expected packaging efficiency, which is this axis, but it has really low diversity. And that's because there's a trade-off. And so the NNK library has really great diversity, but it has crap packaging efficiency. And that's where I'm losing most of my library. Over here, I have a library that's fantastic packaging efficiency, but it has no diversity, because what is it doing? It's taking the one sequence that packages best, and it's making the whole library look just like that sequence, basically. And so we know we probably want to operate somewhere in between those two extremes. And in this case, we were pretty lucky. So in these trade-off curves, if you have a vertical position, so this machine learning D3 is essentially vertically above the NNK library. What that means is I don't take any hit in the diversity, but I have vastly better packaging efficiency. So this is a no-brainer win over the other library. What we don't know is, it possible this library is better? Like, we don't generally know where we want to operate on such a curve. All we know is in vertical lines, the top point is better. So we decided on heuristics of sort of the kink here, and then some other heuristic. We just picked these 3 libraries, and we measured the full titer of these libraries in the lab and checked that indeed they have the kind of behavior we expect to see. And then through an experiment and sort of a decision-making process, we picked this D2 to go forward with, and that's because after packaging it and checking the variants, it had the most diversity of variants post-packaging. And so the idea was, now we've designed a new library that's meant to replace this uniform NNK library, that it will be better for any downstream engineering campaign for a property I've not even thought about in designing this library, just because I'm keeping more of my lottery tickets tickets in the sense that I have a diverse library, but almost things are more likely to package, and I'm less likely to lose those tickets. And if I have more lottery tickets, I'm more likely to win the game. And so as a proof of principle that, in fact, this was true, we took a protein engineering task that the model and our procedure and our library knew nothing about. And the task was to infect non-human primate brain cells, many of which are very hard to infect. And what we showed is that even though our model knew nothing about the brain or these brain cells or anything, that just because we had sort of a better set of lottery tickets, that we had a much, much higher probability of infecting those brain cells than the library that they would have used. And now there's a bunch of follow-ups, but it's not on preprint or anything yet, but we're using this type of thinking to keep going further and further to make these AAVs more suitable for therapeutics, in particular in the brain and on microglial cells. So I think in the interest of time, I'm going to skip over this, but I'm going to just make it a teaser. And so what I'm going to say is I said that the whole thing started by building a supervised model over the 7 mer amino acids, which represent the 21 nucleotide insert, to what this is technically is this log enrichment. So, okay, who's computed a log enrichment? All right, fewer people than who knew Bayes' rule. I'm very surprised by you guys. So this is a standard way to deal with sequencing data, is you have some sequence of interest, and you put it through a select X, and then it has some amount in the starting library, and you count how many times it happened. You put it through a selection, for example, packaging selection, and then you get another account of how many times it happened. And then you compute this like very simple log enrichment, which is really just the ratio of how many times it was after the selection, before the selection, just normalized by total read count. So this is like a very widely used statistical estimate to understand which sequences are more likely to package or pass any selection at all outside of protein engineering. This is a very, very kind of widely used idea. And so this is actually what we did to build these supervised models. But in some other projects, we realized this didn't work. And so I'm not going to go into it. But basically, in the AAV, these were 21 nucleotides, and we could just count how many times they happened. But imagine that this was a really big region, and all you had were short reads. Now you can't count, and you can't just get a log enrichment, and you have a problem. And so basically, if you're interested, go check out this paper, which shows you how to handle a variety of situations you can't natively handle. But in addition, even in the ones you can handle, it just has better statistical power. So I'm going to stop there and leave some time for questions, but maybe I'll just yeah, and I'll leave this up. If you want, you can ask questions about it or read it, and otherwise I'll just take some questions now. Thank you.
Thank you, Jennifer. So I request you to come down at the microphone if you have questions. So we have 10 minutes for questions. Please come down and ask, and we have also online moderator for the questions.
Ask, how do you measure the edge of data? The slide you showed with
measure the what?
The edge of data.
Edge?
Yeah, you mentioned that you use machine learning and physics-based approach in a slide to combine them, where the physics-based.
Oh, I see. Yeah, so that's, I think let me rephrase the question. I think you're saying I mentioned in one of the things I didn't have a whole set of slides for, that Hunter had a project in coherently blending biophysics-based models with machine learning models, where the idea is you want the machine learning model to gracefully back off to the biophysics-based model. And you ask exactly the correct question is, how do you know where to start trusting one and not trusting the other, more relatively speaking? And that is sort of the crux of the problem. And that problem requires that you have a decent handle on uncertainty estimates, and which is also quite a technical area. And I would say this is not solved, but there's reasonable things you can do. And so uncertainty, in a lot of classical statistical modeling, you always just think of sensor noise. You build a linear regression, and there's like an error term, actually, which most people forget about, but there is. And you think of that as just like the error of the machine from which you got the data. And that's like the standard type of noise we think about in statistical modeling. But but in these kinds of problems where you have the issue of extrapolation, there's something that's sometimes called epistemic uncertainty. And that's not uncertainty from sensor noise in your data. It's uncertainty because you're farther away from the training data. It's like absence of knowledge about that part of the space. And so there are certain classes of modeling techniques like Gaussian process regression, which can be quite good at that in many cases. In the modern era, we often take an ensemble of neural networks, and there's arguments about why that works or not. These are imperfect, and that is the key thing you need in order to do this, as well as an uncertainty estimate of some kind for the biophysics-based model, although that one you can handle if you don't have it through some kind of slightly clever but simple thinking.
Hey, Jennifer. Thank you for the really, really amazing and interesting talk.
Thank you.
I was curious, so you presented a slide on basically 3 different ways of looking at this problem of going from P of X to, or P of, maybe P of Y conditioned on X. And you boxed like basically the freezing approach of taking freezing your model and then training something on top of it. Which was interesting.
So just to be clear, we're not training really something on top of it.
Yeah.
We're training actually quite separately. Like they're not trained, I guess they're a little bit trained together through the noise schedule, but yeah. Sorry to interrupt,
Yeah, okay. I mean, maybe it pushes away from my question then. So my question would have been if there was a reason to take the same model or to have the same initial model that's frozen that we're training off of. So, yeah, now I'm...
Like, are you saying what's the advantage in doing that?
So, like, the advantages, so, like, the advantages I could think of are that, I mean, we could save memory and training cost by reusing the model versus with the fine-tuning models, like I've seen some better performances with some of the fine-tuning approaches.
I see. Well, first of all, I wouldn't believe anything you see almost anywhere until you do it for your problem yourself in a way that you are honest with yourself. But I'm sure, I have no doubt that each of those methods has a place where it is genuinely best. And I don't think, and I think, I keep thinking I want to write something, a paper about this, but I have not done that because I haven't done the work to do it. But I think I think John Ingraham in the Chroma paper makes a really nice point about this. So he puts this, he has this crazy, in a sense, super well thought out model to do, to generate backbone structures, so the 3D coordinates of each C alpha carbon. And he puts all this domain knowledge in about geometry and like a prior of collapsed polymers and and again, doesn't matter if you understand, but he puts all this domain specific knowledge into that model with some understanding of what he's modeling, which is protein structures that live in a 3D physical space that represent proteins. And so he can put all this amount of thinking into there, and now he has this model. And when he's doing that, he may not know what downstream use cases he wants, but because he puts so much energy and thought into that model, it's very nice if he can repurpose that model for all these kind of other downstream tasks where he wants to condition on different things. And he can now he has this extremely powerful thing that he can play with Lego blocks by kind of plug and play. And so I think it's true, he could now just take all that machinery and thinking and train it kind of all together as well. But I think in many cases, these models are kind of expensive, and even in industry, people are taxed for compute and so forth. So I think it is like a practical issue. You could, in fact, just redo everything, but I think it's nice not to have to retrain giant models, even if you're just doing some low-rank approximation, blah, blah, blah. Yeah.
Thank you.
Hi, Jennifer. So actually, sort of related to that question. So one thing, I was looking at that slide where you're showing this classifier guidance. One thing that was confusing to me, is it that your unconditional model has to be a diffusion model, or could it be is this kind of a general thing?
That's a good question as well. So that paper is focused on flow matching and diffusion models. And I don't want to, let's say my students are now working on more general versions of that, and I don't want to say it because I don't want them to get scooped. But there are some more general concepts there, and some of the seeds of that exist in some literature. But that paper is specific to well, I would say it's anchored in diffusion and flow matching models.
Okay.
And but basically, if you have any model for which the underlying process is a continuous time Markov process, which is defined essentially by the rate matrix, then all the math they did applies to updating the rate matrix from an unconditional rate matrix to a conditioned rate matrix. And so in principle, there could be a broader set of things based on continuous time Markov processes for which the exact same thing would hold. And then, so that's like a direct generalization of turning the same machinery. And then there's just a more general idea of not exactly using that machinery, but this kind of idea, let's say.
So just to clarify, so then the classifier or whatever predictive model you're creating has to be in the same, it has to be a model which is like kind of, it's a diffusion model prediction task? Or you're...
Oh, you mean the two parts you're plugging and playing there? Yeah, so in that work, that's right. They're both, it's basically saying, I assume the overall model is a continuous time Markov process defined by a rate matrix. Now tell me how to go from a rate matrix, which represents the unconditional model, to another rate matrix, which represents the conditioned model. And it's so all the machinery is converting rate matrices, basically. And that's the requirement for that piece of work.
Okay,
Yeah.
thanks.
More questions? And we can bring up the microphone if you're sitting in the middle and cannot walk down. Okay, then I have a more conceptual question, like I just want to understand from you. So you mentioned how powerful the diffusion models are. So before the diffusion models existed, the generative models...
To be clear, I said they're very powerful for real valued spaces, and I don't think there's clear evidence yet that they're powerful on discrete state spaces. And I have been skeptical, but I now think they are, but it's a very nuanced topic.
Yeah. So would you be, even on the continuous space, could you give us an idea, some conceptual or philosophical idea of why diffusion model is better compared to the
that's a question
method?
I have been asking a lot of people. Like, I have a vague intuition, but I don't have a great answer, and also I don't work in those spaces. And like I'd say, the big wins are in computer vision, and so the people who maybe deeply understand it are in computer vision, which I'm not. But I have the sense that there's something about when you do in most standard generative models, you either somehow just generate the whole image at once in the images, or but something about in diffusion, you're taking like a random image and then you're perturbing it a tiny amount over and over and over. And so there's basically, it's doing something that no other models do, which is touching all the pixels at once, but just a tiny little bit. And so I don't think that's a great answer, but that is like something that those models do that other models don't, is they're touching everything, but just a little bit and doing it over and over. I don't think that's a great answer, though.
Yeah, it's like incremental learning somehow, like from a very random, like if I want to put it in a philosophical concept, like if I want to think about something, a model that doesn't know anything and has some noise and starts shaping the data maybe. And I also think a lot about what's the philosophy behind that makes those models so powerful. That was the question. But I think that is true. It's incremental learning. And yeah, maybe we'll talk offline more.
Okay.
Thank you for that question. Okay, so if there is no more question, then let's thank Jennifer one more time.